This is lecture 5, where we will go over the prokaryotes. Prokaryotes are considered to be single-celled organisms that can inhabit a wide variety of habitats. Prokaryotes first originated in the fossil record approximately 3.5 billion years ago. There are many adaptations that make prokaryotes so successful. Because of their small size, they have a rapid reproductive rate. This rapid reproductive rate also leads to a large amount of mutations, which provides a platform for a high level of genetic diversity, also enabling rapid evolution to occur. Prokaryotes can also have very diverse adaptations that make them highly adaptable to their environments, even in the most extreme conditions. Prokaryotes are currently represented by two domains, the domain Archaea as well as the domain Bacteria. Please take a look to the diagram to the right, where it gives you an overview of the diversity of those particular domains, as well as an introduction to the domain Eukarya as well. Prokaryotes range in size, but they are relatively small, ranging from 0.5 to 5 micrometers. They are considered to be much smaller than their eukaryotic cell counterparts. There are three main types of prokaryotes, and they can be classified according to their shape: spherical, rod-shaped, and spiral. Refer to the diagram below for an overview of these shape descriptions. Prokaryotes also have cell walls. These cell walls are important to maintain their shape and also to protect the cell themselves. Bacterial cell walls are typically made of sugars attached to short polypeptide chains. We can call those peptidoglycans. Eukaryotic cells, on the other hand, have a cell wall that's primarily made of chitin as well as cellulose. In addition, Archaea cell walls are typically made of sugars and proteins. The Gram stain is an important test that can be used to classify bacteria. This is based on properties of their cell wall. Gram-positive bacteria will stain with the Gram stain because their peptidoglycan layer is exposed. Gram-negative bacteria does not keep the stain, and this is because they have a lipopolysaccharide outer membrane which surrounds a peptidoglycan layer. The Gram stain can be used as an important diagnostic tool for both diagnosis and treatment plans. Gram-negative strains tend to be more resistant to antibiotics, and they can also be toxic due to the lipopolysaccharide layer. Some Gram-positive bacteria, however, can also become antibiotic resistant, such as the MRSA strains. Many antibacterial drugs that may be prescribed are aimed to target the bacterial cell wall. This is important because they will not cause damage to eukaryotic cells since the cell wall is not present. Prokaryotes can have many cell surface adaptations. For instance, many prokaryotes have what is termed a capsule surrounding the cell wall. This capsule consists of a layer of polysaccharide and protein. This capsule can create a slime-like layer that serves to do many things, such as attach cells to a substrate as well as each other, attach bacterial cells to an organism, or to protect the bacteria from the host's own immune system. Many prokaryotes can also create endospores. This is when bacteria copy out their genome and can enclose the genome in a multi-layered structure to protect it from harsh conditions. As the cell lyses due to adverse conditions, the endospore is released and remains dormant until optimal conditions return. Many of these endospores are highly resistant to heat, pH changes, as well as dehydration. Many prokaryotes can develop fimbriae to help with attachment. These are hair-like appendages that extend from the cell surface that allow for the bacteria to easily attach to other surfaces as well as each other. Note that these are not the same as pili, which are much longer, fewer in number, and is primarily used to exchange DNA between bacterial cells. Approximately half of the prokaryotes are capable of taxis, which means that they can detect and move toward or away from a stimulus. This movement is primarily due to the presence of one or two flagellum. This can be positive if they are moving towards a stimulus or negative if they are moving away. This can be based on many different stimuli, such as chemicals in chemotaxis, light in phototaxis. It is also important to note that bacterial, archaeal, and eukaryotic flagella are all unique in structure. This indicates that the presence of the flagellum arose independently in evolution. We are now going to take a look at a typical bacterial flagellum. Note that the flagellum consists of a motor, hook that extends from the cell surface, and then the filament that extends out from the bacterial cell. Note that the motor penetrates through both the cell wall and plasma membrane as well as the peptidoglycan layer. There is a rod that is in the center of the motor that also extends throughout the rest of the flagellum. Together, the rod will rotate through the hook, and that will give rise to movement within the filament region. The bacterial flagellum is comprised of 42 proteins that together form the motor, hook, and filament structures. It is likely that the flagella evolved through what is termed exaptation. This occurs when a structure is adapted for one function, but takes on a new role through natural selection and descent with modification. In this instance, it's proposed that the flagellum originated from proteins that were originally used for a secretory system, and they were later adapted for a motor function. We're now going to take a look at the internal organization of prokaryotes. Note that in general, prokaryotes lack what's termed compartmentalization. This means that we do not see the highly organized structure that we do in eukaryotic cells and the division of labor into organelles. Some prokaryotes do have specialized membranes that can carry out certain metabolic functions. In some instances, infoldings of the plasma membrane can have different proteins and varied structure responsible for respiration as well as photosynthesis. Take a look at the diagram to the right to see the infoldings of the membrane responsible for respiration as well as for photosynthesis. The prokaryotic genome is considered to be relatively simple and consists of one circular chromosome. However, it may also contain plasmids, which are small ring-shaped self-replicating portions of DNA, which can be depicted in the diagram below. The genome is typically located in the nucleoid region. However, this is not to be confused with the eukaryotic nucleus, which is completely enclosed in eukaryotic cells.